Hello again. The good news about cellular respiration is, now that we've gone through all of the horrible details for glucose, there are only a few more details to cover in order to get the other macronutrients into the system. Probably the easiest to understand, with the fewest horrible details, is fatty acid beta oxidation. Start with a fatty acid, in this case caprylic acid, stick it onto the end of coenzyme A, and call that acetyl-CoA. No, call that acyl-CoA. Oh. That sure looks like acetyl-CoA, though. It's just a little bit longer. Beta oxidation is going to chop the acyl-CoA into several molecules of acetyl-CoA. It's called beta oxidation because we're going to oxidize the carbon atom that's 2 spots down from the existing carbonyl carbon. It's also called beta oxidation because scientists like to pretend that we all still speak Latin and Greek. Every round of beta oxidation gives us more good stuff. In this example, chopping caprylic acid into 4 molecules of acetyl-CoA will also give us 3 pairs of high-energy electrons on FADH2 and another 3 pairs of high-energy electrons on NADH plus H+. When breaking down a triglyceride or a phospholipid gives us a glycerol molecule, 2 steps, we'll turn glycerol into dihydroxyacetone phosphate, which can then hop right into glycolysis at step 5. Metabolizing amino acids is a bit more complicated because we have so many different amino acids to worry about. The first thing we have to do is get rid of the amino group. For now, let's oversimplify things and just look at the deamination of an amino acid as 2 steps, the first of which hydrolyzes off the amino group, and the second of which oxidizes the number 2 carbon to make it a keto acid. Some amino acids, like alanine and glutamic acid, are called glucogenic amino acids because their deamination turns into things like pyruvic acid and alpha-ketogluteric acid that are intermediates in glycolysis or in the citric acid cycle. Some amino acids, like leucine and lysine, are called ketogenic because deamination turns them into acetyl-CoA and or acetoacetyl-CoA. There are many horrible details, but leucine loses its number 1 carbon as carbon dioxide, but then later on picks up a number 7 carbon from a different molecule of carbon dioxide. The catabolism of lysine is not quite as confusing, but lysine loses both its number 1 and number 6 carbons as carbon dioxide along the way. If we go through all of the horrible details for each of the different amino acids, and we won't, we find that isoleucine, phenylalanine, tryptophan, and tyrosine are both glucogenic and ketogenic. Leucine and lysine are purely ketogenic, and the other 14 amino acids that we use to make proteins are purely glucogenic. For nucleic acids, you don't have to worry about counting calories from nucleic acids when you're taking a nutrition class, because there just aren't that many grams of nucleic acid in a typical cheeseburger. Here are a few more details. The ribose and deoxyribose can be converted into glucose by the liver, just like any other random monosaccharide leftovers can be converted into glucose. We generally don't recycle a lot of the nucleobases that we get in our diet. We pretty much just make our own nucleobases from scratch every time. That probably makes sense. You shouldn't install a used water pump in your car, and you shouldn't use secondhand nucleic acids in your cells. The nitrogen atoms in guanosine and adenosine, the purine nucleosides, get disposed of as uric acid and ammonia. For the pyrimidine nucleosides, cytidine, thymidine, and uridine, the nitrogen atoms get thrown away as urea plus ammonia. For a plain old ordinary cell doing its all day, every day, plain old ordinary cellular respiration, any monosaccharides that we have lying around get turned into glucose and tossed into glycolysis. Fatty acids get chopped up by beta oxidation and are tossed into the citric acid cycle as acetyl-CoA. For amino acids, it's complicated. We just talked about that. Some are glucogenic, some are ketogenic, some are both. Now, if we're running away from a tiger and our leg muscles have gone anaerobic, then we have a problem. Okay, a second problem. We absolutely can't do the electron transport chain without oxygen, and when that shuts down, the citric acid cycle shuts down. With no citric acid cycle, the acetyl-CoA accumulates, and everything else starts shutting down. We might be able to fire up a let's hope we don't get eaten by a tiger kind of homeostatic control mechanism, and some of our skeletal muscle cells, the fast glycolytic muscle cells, otherwise known as the fast twitch muscle cells, otherwise known as the white meat if you're a chicken, these cells can turn pyruvate into lactate through anaerobic fermentation, and then throw the lactate out of the cell. This can keep glycolysis going because pyruvate isn't accumulating. We can make a little bit of ATP in the absence of oxygen. That doesn't necessarily get you away from the tiger, but 2 ATPs per glucose is better than 0 ATPs per glucose. Anaerobic fermentation will only be useful if these cells have a glycogen savings account, because we'll be burning through a lot of glucose if we have to keep this up for more than a fraction of a second. Well then, let's say we got away. Maybe the tiger ate Carl instead of us. Then the liver can clean up the mess we just made, taking the lactate and or pyruvate and or miscellaneous leftover metabolic intermediates of other kinds, and turn all of that junk back into glucose through gluconeogenesis. On the other hand, let's say we're not running away from a tiger. Suppose we're fat, dumb, and happy, and we have more nutrients than we need right now. There's only so much glycogen we can make, but let's get the glycogen savings account filled back up. We can keep turning glucose into acetyl-CoA, but any acetyl-CoA that doesn't get tossed into the citric acid cycle will be turned into fatty acids, storing today's surplus of food energy as the body fat that we can use when we're starving next winter. Or we can just keep getting fatter. Either way. That's enough for now. Thanks for listening. I'll talk to you all later.